Well, good afternoon, everyone. It's a pleasure to see you all. We are basking in a little bit of daylight now that the clocks have moved forward. I hope you all recovered. We were just talking about how 1 hour can actually throw you completely off. It's amazing, but it's a pleasure to be with you all this afternoon, and we are very excited to welcome you to our Science Cafe, to welcome Dr. Ed Benz to be with us this evening to tell us some a very exciting story, I guess I would say, about the power of genetic therapies. And we are very pleased to have him with us. As I look down through the list here, I think he is probably pretty well known to many of you. But for those who may not know, Dr. Benz is the president and CEO emeritus of Dana-Farber Cancer Institute. He is a principal investigator there and holds the Richard and Susan Smith Distinguished Professor of Medicine, Professor of Pediatrics, and Professor of Genetics at Harvard Medical School. He also happens to be a long-standing visiting scientist at the MDI Biological Laboratory, a member of our board of trustees, and currently is the vice chair of that board. So we are thrilled to have you with us this evening, Ed. And just by way of background, I want to tell everybody a little bit about his professional interests. Dr. Benz is a hematologist by training, and so he has spent his career working to understand the molecular mechanics of blood disease. And because of that training, in 2018, when Dr. Francis Collins at the National Institutes of Health launched an effort to speed the development for cures for sickle cell disease, he contacted Dr. Benz and asked him if he would head that initiative. And tonight, Dr. Benz is going to share with us a really wonderful story of the power of genetic therapies. And so it's my pleasure to welcome him. And then I think Fred Bever, my colleague, is going to kind of MC tonight's event. But we wanted to just remind you, we're going to hold questions until the end tonight. If you are afraid you might forget your question, you can easily type it in the chat and we will hold it to the end. Otherwise, you can feel free to use the raise hand feature and we'll unmute you at the end and you can ask your question yourself. So with that, Dr. Benz, welcome. We are thrilled to have you.
Well, thanks so much, Jerry, and thank you all for joining us this evening. Peggy and I regard the MDI Biolab as a second academic and professional and personal home for all of us. We have a place on the island and have been coming up here for over 40 years and made many, many fast friendships. And the lab was always my summer place where I could catch up on my research, which I had trouble keeping up with during my administrative job. So it's a very special place. And if you haven't been there to visit it, please come and see it. The work that's going on there is really very, very special. Now this comes the moment of truth when I find out if share screen works. So let's see if that
Ta-da.
got one more thing to try. My laser pointer. Good. So it's in there. So what I hope to do tonight, and try to do it in about 40 minutes or so, so we have some time for question and answer, is just tell you about this very exciting development, which has been harnessing the emerging technologies for manipulating genes in the form of gene therapies and gene editing. In other words, trying to use genes as medicines and to treat a devastating disease that I've worked on in my own research over the years and taken care of many, many patients with this. I have many investigators and caregivers over the course of the last many decades, the sickle cell anemia. Now, to do that, we're going to have to pull together several threads. We're going to need to know something about how genes work. We're going to need to know something about what sickle cell anemia is and the red cells and hemoglobins that the genetic inherited mutation affects. And then finally, some introduction to the methods of gene therapy and pull it all together to see how these treatments were done. So our agenda for tonight is to cover all of that. So why is there so much excitement about gene therapy? Why do we need gene therapy? Why couldn't we just find medicines to treat all these inherited diseases? And the fact of the matter is that for a couple of reasons, there really isn't much of an alternative for most inherited diseases, other than going after its root cause, which is the defective gene. These are individually pretty rare diseases. Here's some of the most common: cystic fibrosis, sickle cell, what we're going to cover tonight, and Duchenne's muscular dystrophy. You can see below here, they're quite uncommon, but these are the most common of the genetic diseases. So individually, there's not much incentive for the pharmaceutical industry to develop therapies for these patients because there are so few of them. There are also diseases that, because they have intricate abnormalities of the root cause underlying gene, they're not easily targeted with the usual kinds of drug development strategies that we have available to us today. So the solution is to go over after the root cause. And just in case it seems like these are just a few patients, if you add up all of the patients who have the known inherited diseases, it comes to about 25 million Americans. About 1 out of every 14 Americans in one way or another is affected by these diseases. So this is a major medical problem. And now that we have the ability to manipulate genes therapeutically, it's really important that we figure out how to apply this to all of these many other diseases. Whoops, I've already made my first mistake here. Thank you. So this is our agenda for this evening. We need to know a little bit about genes and what they are. So a little bit of skip over biology 101. And then talk about sickle cell anemia, which can only be understood for what it is and how it might be treated with gene therapy. If we talk a little bit about red cells and hemoglobin, the cells and the protein molecules in which this abnormal mutation has its effect, talk about what goes wrong in sickle cell, and then wrap it up by talking about gene therapy and gene editing and how we can bring all those threads of information together to find an effective treatment for these patients. Then we'll conclude with some final thoughts about the implications for these patients and more broadly for the field and for medicine in general. So let's talk about genes for a minute, with apologies to those of you who know this very well, and this is old information. But genes are basically chemicals. They're sequences of DNA, and DNA is a long polymer that has no branches in it. It's just a long stretch in our cells. If you took all the DNA out of it and put it end to end, it would go around the world a couple of times. It's a remarkable molecule. And it has this sugar phosphate backbone. But the important parts of it for us tonight, for our information and the informational parts of the molecule, are these 4 so-called nitrogenous bases: adenine, thymine, cytosine, and guanine, always referred to as their code letters, A, T, C, and G, which, because one molecule, if you see 2 of these molecules are fat, adenine and guanine, and 2, cytosine and thymine, are skinny molecules. So if you want to fit them together in the famous double helix of DNA, you can only have a fat molecule and a skinny molecule bonding together. Otherwise, you'd have bulges and gaps in the way that these things are held together. So those rules that an adenine can only bind to a thymine and a cytosine can only bind to a guanine are the so-called Watson-Crick rules of base pairing, and that's where the information content of DNA is. So if you know the sequence of bases along one strand, it automatically predicts the sequence of bases along the other strand, as you can sort of see here if you wanted to do the counting. But also importantly, if you want to make a copy of this molecule into a molecule that we are all familiar with today, thanks to COVID, messenger RNA, that the sequence of bases in the DNA strand dictate what the sequence of bases will be in the messenger RNA strand. So what you want to encode in the DNA can be carried out of its nucleus and into the cell to be expressed. And as I think you well know, the genes that we care about tonight are expressed in the form of dictating what the amino acid sequence of a protein is through this intermediate of mRNA. So the gene is copied into messenger RNA, goes through a lot of processing steps, and gets trimmed up nicely in the nucleus, and then is released into the cytoplasm, where the structures called ribosomes actually read the sequence copied from the DNA gene like a ticker tape and spool off, as they're doing that, the sequence of amino acids that the DNA sequence specifies. And that unique sequence is what gives these proteins their structural and their physiologic properties. In this example, I'm using beta globin, a part of the hemoglobin molecule that we'll be paying a lot of attention to tonight. And in the aggregate, the protein products of these genes really determine the behavior of our cells and the tissues and our organs, and ultimately us. So we want to keep in mind as we go forward tonight that it is the root cause of these genetic diseases, and in particular sickle cell anemia, will be changes in these code letters that then cause a difference in the kind of proteins that are made at the end of the day. For leaving our quick run through molecular biology, you might have the question of, well, every cell inherits the same genes. So how come red cells make hemoglobin, skin cells make keratin, liver cells make all kinds of enzymes and stuff? How does the cell know which genes to express and which ones not? And this will be important, as you'll see later on, for how at least one of these therapies for sickle cell anemia was made possible. And the answer to that question is that in the vicinity of these genes, around and sometimes even within them, are these different sequences, DNA sequences that I've shown with these boxes here, E for enhancer, S for silencer sequences, that are signals to turn the gene on or off in a particular cell type. So in a developing red cell, for example, there are proteins, so-called transcription factors, that will bind to these regions and either turn the cell, the synthesis of that hemoglobin, on or off. So you have these control sequences that are very important in that they determine the regulated expression of only the right genes in the right cells and at the right times. So we've known those facts about the molecular genetics for decades now. The first discovery of how DNA worked was in 1953. The genetic code was worked out in the late 50s and early 60s. And at the time when I entered molecular biology and as a college freshman in 1964, hot off the presses were a series of papers that showed how those regulatory sequences could regulate the behavior of other genes. And so we could understand how we inherit all of the same genes, but our different cells and tissues make very specialized use of those genes and express only a few. in each cell type. Knowing all that was great, and everybody thought it would be pretty quick work to begin to use genes in the way we're finally able to talk about tonight. But several decades went by, and painstakingly, a few of us in the field could isolate one gene or another and could do things with it experimentally, but never really use it as therapy. And that's because DNA is not that great a molecule to work with by old-fashioned conventional biochemical techniques. But in 2000, there was this momentous event, and I think if we were to look at biology a century from now, maybe considered the most momentous event in life sciences, and that was the completion of the Human Genome Project, which did two things very relevant to us tonight. The first is we now know, as a result of the sequencing of the human genome, we now know the DNA sequence of every human gene, and this greatly facilitates our finding what the abnormal gene is for any genetic disease. And the second is much like the moonshot in the 1960s, provided all kinds of spin-offs of materials and tools that really have changed the way we live today. The tools that were developed to do all this DNA sequencing also are very useful in our research. So tools spun off from the Human Genome Project now make it pretty, I won't say trivial, but pretty easy to do all kinds of manipulation of genes, including making them candidates to be used for therapy. You can cut and paste genes, you can change them in the test tube, you can mutate them back to normal in the test tube, you can do all kinds of things. And the trick now is to take those genes that you can make as candidate drugs and figure out how and in what circumstances you could actually use them as medicines to treat a specific gene. So as a little spoiler alert here that we're going to follow up on and discuss in a little more detail at the bottom of the slide, which is that we know now that sickle cell anemia and all the terrible symptoms that come from it come from one inherited change or mutation in a single code letter or a nucleotide, a thymine to an adenine, of a gene that codes for adult hemoglobin, the so-called hemoglobin A. And that results in the change of only one amino acid in hemoglobin A, and it's a glutamine to a valine in the beta chain of adult hemoglobin, producing this complex and severe disease. So it's always amazing to me to think that here's one mistake out of 3 billion. Any of you who work in quality control and could get a one error in every 3 billion tries would probably get promoted to CEO. But this one error out of 3 billion bases can cause in this disease a devastating and life-shortening illness. And this is true for many, many other of the genes that come from the 20,000 or so genes that we have that code for proteins. So why is that so? Excuse me, I keep clicking things wrong. So that's what we're going to talk about next is sickle cell disease. How does this disease result from this one error in the 3 billion base pairs of the human genome, and this one abnormal amino acid out of all the proteins that we make? To do that, we have to just spend a quick review of what red cells and hemoglobin are, because that's where this mutation has its consequences, and then talk about how the changes in that molecule and in those red cells goes wrong in sickle cell disease. So talk a bit about just how sickle cell disease was discovered and what people knew about it. That is, in 1910, a doctor named Herrick, who also described heart attacks, myocardial infarctions, he's a cardiologist, took care of a dental student named Walter Noel, who showed up with anemia. He was an African-American student, actually, from Grenada, actually, and he had anemia. But he also had these constant recurring painful crises of joint and muscle, chest and belly pain that would come and go. And what Herrick noticed that nobody had ever noticed before in patients like Mr. Noel was that his blood had these very abnormal shaped cells here that he called appropriately sickle cells. Subsequently, people found out that this disease occurred almost exclusively in people of African descent, that it was inherited, and it is quite common, actually. The sickle cell trait, where you only inherit one abnormal gene out of your pair of genes, was present about 8% in the population. So that's a lot of people. Fortunately, you only get sick if you inherit one gene, the sickle cell gene, from both parents, so that both of your beta globin genes are sickle cells. It's much less common to have the disease than 8%, but it's still thousands of patients, about 150,000 patients in the United States have sickle cell anemia. Years went by when people didn't quite know why these cells sickled the way they did, when someone discovered that if you put these patients' blood in a high oxygen environment, they look fairly normal. But if you took all the oxygen out of their environment, so this would be like being in venous blood versus arterial blood, the cells sickled, as you can see from this slide here. So there was something about going on in the red cell that caused these cells to change shape when they were passing into the venous circulation. And it took, again, many years before it was discovered by Linus Pauling and Harvey Altano back in 1951. So this is, 20-some years going by, that this sickling phenomenon was actually due to the inheritance of an abnormal hemoglobin. They were able to show that this hemoglobin could be isolated from normal hemoglobin, and they called it sickle hemoglobin or hemoglobin S. Then a few years later, Vernon Ingram discovered that the problem with this hemoglobin S was in one subunit of the hemoglobin, as we're going to see in just a moment. It's called the beta chain, and that in the amino acid at position 6 of that beta chain had the chains that I've already mentioned to you of beta 6 going from glutamine to valine. And then they could show that that caused this sickling phenomenon, and we'll get to that in just a second. So what about hemoglobin? We know now that that's what molecules is abnormal in sickle cell anemia. It's a fairly complicated molecule, but it's very essential to life. It's what makes red blood red, but that's because it binds oxygen in the lungs, where it keeps the hemoglobin red. It holds onto it as it goes through our blood vessels, and then in the thin-walled capillaries that feed all of our tissues with a blood supply, it gives up the oxygen and delivers it to the tissues. And the way it works is that it's a complicated protein of tetramers, of four protein subunits, each of which enfolds a heme molecule, and inside the heme molecule is a single atom of iron. Iron loves to bind hemoglobin, but it doesn't like to give it up. And the purpose of all this other stuff surrounding the iron is to configure things chemically so that in the high oxygen environment of the lungs, hemoglobin acquires the oxygen, but in the lower environment of the tissues, that the hemoglobin will actually give it up. It will cause the iron to give up the molecule. So it's this reversible pickup and delivery of oxygen that makes hemoglobin essential to life. The important point for us tonight is that we make hemoglobins at different times of our lives that have different compositions. We can skip the embryo, but fetal hemoglobin, or hemoglobin F, has alpha chains and gamma chains, and adult hemoglobin, the major one that we care about tonight, has alpha chains and beta chains, and that these go through an orderly switch, as you can see in the slide on the right around the time of birth. So we live off fetal hemoglobin during gestation, And then right around the time of birth, we switched to adult hemoglobin. The important point for us tonight, from the point of view of therapy for sickle cell anemia, is that sickle cell anemia patients are born well because they're living off fetal hemoglobin and not off adult hemoglobin. It's only when the switch occurs later in the first year of life that they begin to develop the symptoms of sickle cell anemia. So one desirable way potentially to treat sickle cell anemia would be to prevent the switch. We know you can live off fetal hemoglobin as an adult, but trying to figure out how to do this is the challenge. And you're going to see that in one way or another, directly or indirectly, this is what the gene therapies that we're going to talk about are made of, is that actually trying to have the sickle cell patient make more fetal hemoglobin or an engineered hemoglobin that's very much like fetal hemoglobin. So hemoglobin is packaged in red cells. If I were to inject you with a syringe full of hemoglobin, the hemoglobin would be poured out in the kidneys in just a matter of minutes. So we'd be making pounds and pounds of hemoglobin every day if we just had it floating around in the circulation. Instead, it's packaged into red cells, as you know, and red cells are Uber Eats or DoorDash delivery trucks for all that oxygen. And they contain 90% of their protein in the form of hemoglobin. And that's really their main function in the circulation. They make about 1,000 trips a day through the bloodstream, but they wear out after about 120 days, after they have about 120,000 miles of wear and tear, and they're removed. That means we have to replace about 300 billion red cells every day. We have about 35 trillion in the body. And the way that these are replaced is from what's called a multipotent hematopoietic stem cell. It gives rise to all of the blood cell types, red cells, white cells, and platelets, as you can see in the slide here on the right. And the way it's done is that there is this so-called hematopoietic stem cell, or HSC, that we'll be talking about from here on out, that actually is the only cell that is permanently present in the bone marrow. All these other cells, when differentiation starts and these cells go down these different pathways, they're all transient. So this cell becomes that cell in red cell development, it becomes the next one, the next one, the next one. And as they do that, they disappear. So they only live for a matter of days or weeks in the bone marrow. It's this HSC that's the permanent cell. So if you want to correct a genetic disease of the blood cells, then what you have to target is this hematopoietic stem cell. And you'll see later on, as we talk about the therapy, that dictates a particular strategy to be able to get at this cell and to modify it with a therapeutic gene. So what goes wrong in sickle cell anemia? Well, as I mentioned, as red cells pass from the artery into these thin wall capillaries, they give up the oxygen to the tissue that they're surrounding. And in that course of that, the hemoglobin actually changed shape from a so-called relaxed to a tense configuration. We don't need to worry about that, except to know that normal hemoglobin tolerates that shape change very well. And it stays in solution, and nothing happens, there's no harm, no foul, and it goes back into the veins to be reoxygenated in the lungs. But in sickle cell anemia, because of that one amino acid change, as the hemoglobin goes through the capillaries and gives up its oxygen, that amino acid change causes the hemoglobin to polymerize, to form these fibers, these needle-like fibers that are called taptoids and are toxic. And they make the red cell change from having an inside that's watery to having an inside that looks like and feels like jello. And they become very stiff and viscous. And because red cells have to squeeze through these very narrow capillaries, these very stiff cells don't make it through. They plug up the capillary, and so downstream, blood flow is impaired, and the tissues and the capillaries become hypoxic. They don't get enough blood flow. We call that in medicine ischemia. So the problem with the hemoglobin S, or sickle hemoglobin, really becomes a problem of blood flow. And that's what causes the symptoms of sickle cell anemia, these clinical consequences. No blood flow, of course, means no oxygen going to these ischemic tissues, no nutrients, sugar and other things being delivered, vitamins. There's no removal of cell waste, all the byproducts of our cell metabolism, and tremendous pain. If anyone's ever had in this audience angina or phlebitis, where you have blocked blood flow, you have a sense of how painful that can be. So what sickle cell patients have are these recurrent painful crises where when sickling happens, they have ischemia in different tissues and feel this enormous pain. But in addition, ischemia, as you can imagine, injures and eventually kills the cells that aren't getting enough blood flow. So you have these microscopic areas throughout the body undergoing microinfarction, or the organs causing the organ to malfunction and eventually to fail. And it's all these different organs, particularly the spleen, kidney, and the eye. You get an encephalopathy. Patients with sickle cell suffer from brain fog. There's a sickle cell heart disease. Lots of damage to bone and joints, and particularly to the lungs, so that these patients get pulmonary hypertension later in life. Plus which, the sickling process damages the red cells. They only live 10 to 20 days in circulation instead of 120. So you have all the symptoms of anemia and all the symptoms resulting from the excessive red cell turnover, like gallstones and leg ulcers. And patients who have these kinds of debilitating conditions get repeated infections. One of my patients told me once, who's suffering from just about all these complications, that we had the wrong name for sickle cell anemia. He said, "I call it sick as hell anemia." And I can tell you that any patient or anyone taking care of these patients will report that this is a devastating lifelong disease. Thanks to the treatments that we have, which are all supportive, patients can now live beyond childhood, which they used to not do, but only about the age of 55. The life expectancy in the United States is about 78, and it's in the high 70s or low 80s throughout the Western world, but not in sub-Saharan Africa, where most patients are. You don't even get to age 55. There's a tremendous amount of death in childhood. So we have therapies for this disorder, but they don't really modify the disease. They're simply coping with the complication. There are a couple partial, imperfect disease-modifying therapies. And one is stem cell transplantation. In other words, could you do a bone marrow transplant like we do for leukemia, where you use chemotherapy to wipe out the patient's stem cells, HSCs, and replace them with normal stem cells from a donor? And there are patients who've been effectively cured with sickle cell anemia by doing this. But the problem is there's a 10% mortality associated with that. So it's a little hard to justify doing it unless you have the most severe forms of this disease. And there are many long-term complications of bone marrow transplant we can't go into today. And it costs about $200,000 or $300,000 to do it. On top of which, in the African-American population, there's always been a challenge to get sufficient numbers of donors who can actually donate genetically compatible bone marrow, immunologically compatible bone marrow. You're only drawing from, in this country, about 18 to 20% of the population. But there's another drug that helps, and we're going to talk about that for a second because it tells us what we could do in gene therapy, called hydroxyurea. So the question is, how could a chemotherapy drug actually help us treat sickle cell anemia? And that comes from observations that began to be made in the 1970s and 80s, that there was a condition called hereditary persistence of fetal hemoglobin, that a number of patients, it's an uncommon but not super rare condition, make a bit more fetal hemoglobin after birth. They're perfectly healthy. And a few of these patients make up to 20 or 25% fetal hemoglobin in adult life, whereas the normal level is around 1%. And some sickle cell patients were observed to have inherited both this HPFH condition and sickle cell anemia. And lo and behold, they had mild disease. And in fact, you could correlate how much fetal hemoglobin they made with the incidence of the major symptom of sickle cell, which is these vaso-occlusive crises. So how come inheriting more fetal hemoglobin gives you milder disease? Well, we know why this is so, and it's two ways. First of all, we know that red cells can only make so much hemoglobin. They can only hold so much hemoglobin inside the red cell. So the more of another hemoglobin, in this case fetal hemoglobin, you make, the less hemoglobin S there'll be. So there's a replacement. There's less hemoglobin S in the cell. That means there's less sickling. Even better, hemoglobin F was discovered biochemically to stabilize those toxic tactoids. In other words, it blocks the polymerization. It's an active inhibitor of the polymerization of hemoglobin when you form deoxyhemoglobin S, and that also inhibits the sickling. So you get a double whammy, double benefit from this replacement and active inhibition of sickling. So increasing fetal hemoglobin is a valid therapeutic goal for sickle cell anemia. Could you do that with a drug? Well, the answer is sort of. Another observation made during about the same time is that bone marrow stress, such as people recovering from chemotherapy or people recovering from a bone marrow transplant, when the bone marrow has to be overactive to repopulate all the blood cells in our circulation, that there was a burst of hemoglobin F synthesis when that happened. And that a milder chemotherapy drug, which is in use for other conditions, called hydroxyurea, better tolerated than most chemotherapy, so can actually be given chronically, increases fetal hemoglobin up to 25% in some patients. So someone said, well, let's try that in sickle cell. And sure enough, when it's used in sickle cell patients, it can help some patients significantly. And it's now standard practice to give it a try. It's not a magic bullet. It doesn't work in everybody. People are very variable in how sensitive they are to the drug in terms of turning on fetal hemoglobin. And they're also quite variable in how sensitive they are to some nagging side effects. After all, this is a chemotherapy drug, and it is toxic to cells. So you have to find just the right dose and hope that it will turn on fetal hemoglobin, and you have to take it every day, and sometimes a couple of times a day for a lifetime. So it's imperfect and it's incomplete, but it does give us an important proof of principle, two things that lead us into gene therapy. We can alter the hemoglobin composition of red cells pharmacologically. It's possible. In our case, the pharmacologic agent will be a gene. And even partial increases. You don't have to get complete replacement. If you get above about 20%, you can have a beneficial clinical effect on patients. And it's because of that double whammy. It's not just that you're reducing the amount of sickle hemoglobin, but it's because the hemoglobin F actively blocks the sickling of the remaining sickle hemoglobin inside the red cell. So the goal with both gene therapies I'm about to tell you about was not to edit out and correct that mutation that causes sickle cell anemia. And hopefully by the conclusion of these last few slides, you'll see why that they didn't try to do that, but instead to either turn on fetal hemoglobin in the stem cell or manufacture hemoglobin that has the properties of hemoglobin F, but can still be expressed in adult red cells. So let's ask, if we want to do that, if we want to do that by manipulating a gene, what is gene therapy? Well, it's a technology that manipulates DNA molecules. You want to replace or repress or modify the behavior of the missing or the underactive or dysfunctional gene, in our case, the beta globin gene of hemoglobin, that's responsible for a disease in order to achieve a therapeutic goal. There's two ways to do that now. One is you can add a gene. In other words, you can find a way to manufacture the gene you want to use, put it inside a vector, and get that vector to infect the cell with the gene, and add the gene to the complement of genes being expressed in the target cell. The other way is say, well, that can work, but that, as you'll see, has problems and can get complicated. What if we could have a device that actually went in and modified the genome in some way that created the desired therapeutic effect? So instead of bringing a gene in and incorporating it into our genome, you make a change in the gene as it's sitting in situ on its chromosome. And that is the exciting new frontier of gene therapy. So the critical components of any gene transfer strategy are the therapeutic payload, the transgene or the editing apparatus that needs to be delivered. Then you need a delivery vehicle, and for the most part nowadays, those are engineered viruses where the disease-causing genes in the virus have been removed and replaced by inserting the payload into the genome. Because viruses have their own issues, immunologic reaction to them being one, a lot of work is being done on synthetic delivery agents like lipid nanoparticles, but those are not quite ready for therapy yet. And then finally, you have to know what cell you want to target and how to make these delivery vehicles find that cell specifically. For us, that target is going to be this hematopoietic stem cell, and it cannot be presently targeted inside the body. It's very rare inside the body, and none of these vectors can specifically find it without infecting a lot of other kinds of cells. So we need a different strategy to be able to do the gene therapy. This is what happens at the cellular level with gene therapy. So you have your virus into which you've inserted this new gene. It finds the cells that it's going to bind to, hopefully the ones that you want to target, enters the cell, gets into the nuclear membrane, and then injects its DNA into the nucleus of the cell, where it gets incorporated into the genome and gets expressed. So basically, this is in some ways the simplest part. We know how to get viral particles. We know how to get the DNA injected in. The tricky parts are finding the right cell and engineering the gene so it's only expressed in the right way within that cell. So the two basic strategies is, one would be just inject the gene. And this is what a lot of people think gene therapy is all about. Inject it into the eye if you want to correct congenital blindness. And that's actually been done for the first FDA-approved gene therapy back in 2010. But for us, because we can't just inject directly into the bone marrow, and we can't get the gene all over all hematopoietic stem cells scattered throughout the bone marrow in the body, you use ex vivo therapy. So what you do is you can get hematopoietic stem cells out of the bone marrow. You can mobilize them with a drug as you're going to see in a second, grow them in a petri dish, expand them so you have enough cells to put back in the patient, and then transform them with your vector and re-inject them into the patient. And that's how the first approved gene therapy for sickle cell was done. In this case, the scientists, the Bluebird biotech company, engineered a hemoglobin called P87Q. And what you need to know about this is that it has a change from normal beta globin in the 87th amino acid. And they did that because biochemical experiments had shown that if you make this change in the beta globin gene, it behaves a lot like fetal hemoglobin in being able to inhibit sickling. The advantage is that you can't just put fetal hemoglobin gene into an adult stem cell because it'll be shut down. Remember, the adult stem cell has inhibited fetal hemoglobin. So this takes the advantage that it's on a beta globin gene, which will be highly expressed, but it now has this acquired property of inhibiting sickling. So using a drug called plerixafor, you give the patient who's been transfused to eliminate the symptoms of sickling. You mobilize these stem cells into the bloodstream, isolate them, grow them in a petri dish to expand them, add the virus carrying this engineered gene, put them back into the patient, and allow the bone marrow to repopulate, which requires another 2 or 3 weeks in a hospital. So you can see this is very complicated therapy, which requires the patient participation in the therapy for almost half a year. So they did a phase 3 clinical trial with this, and 30 out of the 32 patients who were treated no longer had severe painful crises requiring hospitalization. In fact, 28 of those patients had none at all, and their blood counts were restored to near normal. This drug got FDA approved after observing these patients for times up to 4 or 5 years, and the therapy was holding. And so it's now on the market, and it's about to enter clinical practice. If you think doing gene therapy is hard, or going through it is even harder, the hardest thing is pronouncing the name of this drug. So I'm going to call it Lyfgenia, which is, you're not supposed to use brand names, but anyone who can say the name of this drug 3 times rapidly gets a tenured professorship at Harvard. The other company that did this, the other therapy, uses what I think is going to be the technique of the future, wherever it can be applied, and that's gene editing. And what gene editing does, as we said, is it actually goes into the human genome and makes a change that gives you the desired therapeutic effect. It alters the properties of the genome in a way that is clinically beneficial to the patient. And this has 3 components. One is a guide RNA, which is what finds the DNA sequence that you want to change. The second is a protein editing complex, and in it is the third component, an endonuclease. So because of the guide RNA, this complex will find the DNA sequence that you want to alter. And the endonuclease will make a cut there, and you can do it. You can make 1 snip just to open it up, stick something in the middle, or you can make 2 snips to take out a big piece of a small piece of DNA. And the rest of this complex helps hold all of these things together so the cut ends don't go flying out into space. And it also brings in our naturally occurring DNA repair mechanisms that help sew all these cut ends together. And by manipulating how you make the cuts, you can bring in different aspects of our DNA repair mechanisms so that this is very flexible therapy. You can remove a gene or part of a gene, you can insert a new one or part of one, or you can alter the DNA sequence of the gene in a level of precision as small as 1 base or up to several dozen bases. So many, many ways you can change the genome quickly and efficiently and safely with gene editing. And the final gene therapy that I'm going to talk about did just that. That changed the way we can actually turn on fetal hemoglobin in an adult stem cell. Now, the way to do this requires I tell you a little bit about what's been discovered about why we don't make fetal hemoglobin in adult life. And I should say this work, and the reason we have this gene therapy is a tribute to the lab of Dr. Stu Orkin, a good friend and colleague of many of us, who just did this incredibly elegant work to find out why we couldn't switch on fetal hemoglobin in adult life, at least not effectively, and figured out how we might take advantage of what they learned to make a therapeutic gene, I should say gene therapy, that actually has been successful. So what they discovered was that there's a protein called BCL11A. And when BCL11A is present in a developing red cell progenitor, it blocks the ability of transcription factors to find the promoter for the gamma globin gene of fetal hemoglobin. So in adult cells, we're expressing this BCL11 gene shown here as off chromosome 2 and causing it to basically blind the transcriptional apparatus in the red cell progenitor from finding the gamma globin gene. In fetal life, there is no BCL11 expressed, and so we can make plenty of fetal hemoglobin. So why not just knock out BCL11A? You could do that with gene editing, just go in and knock out the gene. Well, unfortunately, BCL11A is also very important for lymphocyte development and many other cell type developments. So if you knock this out, and this was done in a mouse, the mouse died. You can't live without BCL11A, at least in the right tissue context. But happily, the Orkin group discovered that in the developing red cell, the expression of BCL11A depends on the action of an enhancer that's only active in red cells, because it's only in red cells that it's bound by the right protein factors. So if you could knock out or edit this enhancer molecule, you could shut off BCL11A only in the developing red cells and not worry about harming any other of the important functions of that gene. And in fact, that's what was done in this gene therapy. BCL11A erythroid enhancer was edited. These 4 bases in bold were knocked out. And so the cells in which this edit occurred don't make BCL11A and will express fetal hemoglobin. And Vertex and CRISPR Therapeutics created a molecular apparatus that did exactly that, edited this erythroid enhancer shown right here. And in clinical trials, they showed that BCL11 expression was greatly diminished only in erythroid progenitors. Hemoglobin production was robust, 30 to 40% in many of the cells. 29 out of 30 patients had no crises for 2 years. Their anemia was improved, the hemolysis was reduced. And that has been FDA approved just at the end of December of this year, and it is now on the market and ready for clinical practice. So those are the 2 gene therapies that have us so excited. So what final thoughts, given those considerations, should we keep in mind as we go to questions and answers? Well, the first is this is great. It's terrific. And if you are a patient who's received this and endured all the complications of that treatment, you have a much better situation than you did going in. You have the hope of something that feels almost like a cure. But along the way, you suffered the risks and complications of chemotherapy. You've had to live through all the time you recover from the bone marrow transplant. and you have been exposed to a genotoxic drug, busulfan, which carries with it a small but not trivial risk, about 1 or 2% or so, of developing myelodysplastic disease, which then evolves into leukemia. So this is not without risk, either of these forms of ex vivo therapy. Then there's access. You got a sense from all that these patients go through and the incredible sophisticated technology, the care in a bone marrow transplant center that are required, that there's only a few places in the country or in the Western world that are capable of administering this therapy. Patients have to be able to basically give up several months of their lives to go through the therapy, and the cost is $2 to $3 million just for the product, plus all the costs of all this other care that's surrounding it. We don't yet know, although therapy is looking good after a few years in these patients, how long it's going to last. And these therapies stop the disease process, but they don't correct any of the end organ damage or psychosocial damage that's been done to the patients before they get the therapy. So they need long-term follow-up and observation care, so-called survivorship resources. And that leaves open the questions, who should get it and at what age? And it's pretty clear that we know that these gene therapies work. We need to find better, simpler, and safer ways to deliver them efficiently so that you don't have to go through all of these complications. But gene therapy is here and it's spreading. And it's already in use for many of these diseases. Leber's amaurosis was congenital blindness was approved in 2010. Now we have sickle cell disease and a related hemoglobin disease called thalassemia that can be treated with these same therapies. Hemophilia gene therapy has been approved in Canada and will almost certainly be approved in the US sometime this year. And many of these others are in active clinical trials, and this is only a partial list. So I'll open up for a few minutes of questions that we have. But to me, the implications of what's been done in sickle cell here and a couple other diseases is that gene therapy is going to be part of how we treat very serious genetic diseases, and that we have to think about what are the issues that we have to deal with, cost, how will we make this accessible more universally, how do we find safer ways, how do we ensure that these therapies will last a lifetime, because it's not at all clear that you can go back in and do redo of the therapy, and how many different ways can we apply it, and potentially to other diseases that are acquired, where manipulating the genome may allow us to get around the problems that create the acquired disease. So thanks very much for your interest in the lab, for joining us tonight. I thank you for your attention and your perseverance in going through this pretty heavy duty stuff. But most of all, let's all thank the patient pioneers who endured these clinical trials in this and many other areas of clinical research and make new therapies possible. So any comments or questions or thoughts in our last couple of minutes are most welcome. Thanks very much.
Thank you, Dr. Benz, so much for such a brilliant leading us through from some very basic concepts to some very complicated, but thanks to you, understandable results. And I think we don't have a lot of time left, and there are some questions starting to be asked in the chat, and I encourage people to put your questions there or just to raise your hand, and we can try to call on you. So just while people sort of get themselves together, Dr. Benz, when we were talking before the cafe started, you mentioned that this is it's an incredible mechanism that's been created, but it is fairly roundabout still. And you mentioned that it is possible to go in and directly correct the mutation. And I think you meant go directly into the stem cell. Can you describe what you meant by that and maybe why we're not doing it yet?
Right. Yeah, and the answer is actually pretty simple, is that as great as these current techniques are, they're not efficient enough to correct all of the sickle genes in all of the stem cells. So some stem cells in that process don't even get infected. In others, the efficiency of knocking it out within the cell is not 100%. So you will reduce the number of sickle cells that are produced, but maybe not enough to make a big difference in the disease. The beauty of fetal hemoglobin is that double whammy. Even if you only get partial efficiency, you also get the active inhibition of sickling. And that's why people turn to either expressing fetal hemoglobin, or in the case of the other gene therapy, engineering a hemoglobin that mimics the inhibitory effect of fetal hemoglobin.
So it is basically you get a regime change, but you don't know how permanent and how complete that regime change is.
Right.
Okay. Barbara's asked whether it's a tuberculosis bacteria that might be used. And Barbara, maybe if you could pipe up to explain your question a little bit more, if you're still with us. You can unmute yourself.
I think
Yes,
so there-
my friend had renal cancer. She was injected with tuberculosis. I'm saying virus, bacteria. I'm sorry, I'm stupid. I don't know what it is. But I've heard also that polio is injected for other kinds of cancer. Is this the same? Are we going into the stem cells? What is this?
So just two different answers to that question. Thank you for the question. For bladder cancer, maybe for renal cancer, but it's mostly used for bladder cancer, is that it's a form of a tuberculosis-like bacteria that our bodies react to with a strong immune response. And it's the immune response that has the anti-tumor effect. So it's not, the virus is not attacking the tumor directly, but it's creating an immunologic response. The nice side effect of that immunologic response is it stimulates the immune system to recognize the tumor. So it's one of the early forms of this now rage that's out there about immunotherapy using other kinds of molecules. In the case of the polio virus, what polio virus does is it infects neurons. So using a modified polio virus that will affect the neurons within a brain tumor, but won't replicate and cause polio in the rest of the body. And they're using the virus just inside the environment of the brain tumor to try to kill it. It looked very promising in small clinical trials and larger clinical trials. It's usable for some patients, but it's not the magic bullet that we hope for.
It can't be accepted by some of them. Yes.
Right.
Yeah.
I'm wondering if people know we might take just 2 more minutes where it's past 6 o'clock now. But I want to make sure that people have had a chance to at least to raise your hands. There's a function in the chat. I see David Dawson has raising his hand. David, please go ahead.
It's my real hand.
Yes.
So Ed, I was intrigued by this phenomenon you described as the sickled cell, and I may not get it right, sort of having to squeeze through at some point, and it becomes, there's some sort of transformation that goes on as if the hemoglobin is polymerizing or something. That sounded to me like sort of a sol gel transformation that the protein chemists talk about.
It is a sol gel transformation. Fortunately, it doesn't happen instantly. It takes time, so not all the cells sickle as they go through, and until they get to the bigger vessel. The diameter of many capillaries is around 3 to 4 microns, and a red cell is diameter is 7.5 microns. So the red cell has to be, on the one hand, very pliable and flexible. It's a biconcave disc. It's kind of like a beach ball. It's only a quarter filled with air. You can squish it and twist it, and but it's also got to be strong enough to go through all those mechanical stresses to go through. When you get the gel inside that instead of a liquid solution, the red cell can't make all those changes, can't be stretched and squashed, and it
Right.
plugs up the capillary, and that's why you get the occlusion, the so-called vaso
Thanks.
occlusion. Now, I work on red cell membrane protein for the last 30 years of my life, David. So if you'd like me to spend 9 hours telling you how the membrane works, I'd be delighted to do it.
I've been having trouble sleeping, so that might be a good idea.
We have one more question that it came in through the chat from David Preston, and forgive me if I don't pronounce all these words correctly, but he said, in general, an autologous donation of bone marrow is ideal. It sounds like, in general, is bone marrow aspiration / biopsy, or are blood stem cells collected by apheresis? The main question is, what type of stem cells from here are ideal to use for such gene therapy editing?
Right. Yeah. So it's a lot. Apheresis, for those not familiar with it, is a process by which you remove blood cells from the body, and then you use a variety of fractionation techniques to pull out the cell, isolate the cell you want. In this case, the stem cells. Nowadays, even for autologous bone marrow transplants, rather than sticking a needle into the bone marrow 20 or 30 times, which is the way we used to get marrow from donors and still do for some purposes, you can use these drugs. The one I mentioned, another one's called GCSF, which cause the bone marrow to release the hematopoietic stem cells into the bloodstream, and then you just draw off blood, and you can isolate the stem cells from the blood that you draw off and give the rest of the blood back to the patient. That's what apheresis means.
Okay,
That's
well,
the way
thank
it's
you.
done.
So there's the answer. It is apheresis. So, Dr. Benz, thank you once again. Thanks so much for all of your time here and for explaining all of this to us. And it's very exciting. I'm sure there are people who have many more questions about where this is all headed, and we look forward to talking about that more in the future. The participants, there will be a recording. This has been recorded, and we'll send that out to those who've registered, and I'll also see a note about it in our upcoming electronic newsletter. And so thanks all of you for attending this. I just want to mention that a couple of more events coming up on April 3rd at 5 o'clock, a special event for alumni and mentors, visiting scientists. It's called MDIBL Moments. And for students or visiting scientists or course participants, it's an opportunity to get together online and talk about that experience in the past and what it's done for everybody, and keep those connections going. On April 8th, the Science Cafe will be online again. Our guest will be Nishad Jasindara, who is giving a presentation titled Climate Change and Pollution, a Double Threat to Kids' Health. He does a lot of work around how climate change can affect toxicity out in the environment and then affect particularly children's health around the world. And so you'll be able to register for that as well. It's the same time on April 8th at 5 p.m. We'll be in touch with you to make sure that you get another reminder that it's happening. So thanks so much to everyone for participating tonight, and we'll look forward to seeing you next time.
It's great. Thanks,
Thank you
everyone.
all. Thanks
Ed.
all for your
Bye.
attention.
Thanks, Ed.